I'm a PhD candidate at biological science under Dr. Lisa Stein and Dr. Dominic Sauvageau. So I'm going to be deviating a bit from the first three talks and talk more about known carbon-based fuels. So I'm developing genetic tools for a bacteria called Methylomicrobium album BG8, which will be a platform for conversion of methane to isoprenoids. Before I go into my research, let's think about summer of 2021. Yes, there was COVID and vaccine, but there was another thing for which Canadians will remember the summer, the extreme heat wave. There was historical heat wave, which the reason for is, as you all know, the climate change, which is again brought about by the anthropogenic greenhouse gas emissions. And here I'm going to talk about one such gas, is methane. So methane is second largest contributors among all the greenhouse gases. And while it's small in amount, it is actually more potent than carbon dioxide. So we should be really careful of, with the methane emissions and trying to find a way to mitigate the emissions. Some of the anthropogenic emission sources are the oil fields during oil productions, as well as during the process called venting or flaring in the oil refineries. You have your landfills and some of the agricultural process, such as rice paddy productions. So one of the way you can mitigate is this using this methane as feedstock for biomanufacturing. Now, how you can produce some valuable stuff from methane? Use a group of bacteria called methanotrophs. So methanotrophs essentially eats methane to produce energy and biomass. So if you have a product and you have the genes for the products, you can engineer this particular bacteria to produce this product. So my product of interest here is isoprenoids. And why isoprenoid is because it's something of an ultra performance fuel precursor. So what is ultra performance fuel? It's the fuels used in like jet fuels and aviation fuels. These are highly valuable products. So as you can see here, in one way, we're trying to find a solution for the greenhouse gas emissions. And in another way, we're getting a valuable product out of it. So what are the challenges for reaching these goals? So while there are multiple challenges, I'm going to talk about something related to my work, which is genetic engineering, where we try to engineer the genes that produce isoprenoid in the bacteria and get more yield out of it. How we do that? What we do is introduce something into the bacteria called a vector or a plasmid. This is a piece of a circular DNA that goes inside the bacteria and controls the expression according to what we want. So, but bacteria do not like taking up this extracellular DNA. So but we have been able to generate a successful protocol for it. So what we do is we take our M album BG8, we take another bacteria that contains a vector, we essentially allow them to mate together, which allows the genetic exchange, and we call this process conjugation. And after that, we have our M album BG8 with the vector. So now we have a way to include the DNA inside the bacteria. What's next? Control the expression. So how we control it? Using something called a promoter. So a promoter is essentially a very short piece of DNA that control expression of a gene. So here we took a gene called green fluorescent protein, and we took a bunch of different promoters. And we see how these promoters control the expression. What you see here is the result of genetic expression studies and the effect of promoters on that expression. You have two promoters that allows a very high level of expression. You have one that is kind of in the low range. So now we have three promoters that allows different expression of the same gene. So that means using a combination of these promoters, we can control the expression of our gene of interest. We have another promoters, which we like to call an inducible promoters. Why? Because in this particular promoters, if you add something called an inducer from outside, you can very tightly control expression. So you have zero, when you have the zero concentration of the inducers, the expression is really, really low or even zero. When you have some significant amount of the inducers, the expression increases and reaches a very high level. So now, as you can see, we have efficient controlling mechanism. We can actually go for our isoprenoids of interest. So these are our ongoing and future reactions. So what we're doing now is we're trying to select an isoprenoid of interest based on something called product toxicity. So in general, in biomanufacturing, you cannot select a product which has some sort of toxic effect. So we have to select something that has least of the toxic effect. We select the genes that produce this isoprenoids, and then what we do is we would try to control the expression of these genes. So essentially, we get some level of production of isoprenoids. Now, what I would like to point out is while I'm developing this manufacturing platform for isoprenoids as a biofuels, in future, using this same tool, somebody can make a biomanufacturing platform for isoprenoids that have other uses, for example, in pharmaceutical industries or dye industries. So with that, I'd like to close my talks, and I'd like to acknowledge my guides, Dr. Lisa Stein and Dr. Dominic Sauvageau, as well as two of our collaborators, Dr. Hennard and Dr. Michael Guanri. They were the one who trained me in the developing genetic tools, and also my lab mates and all our funding partners. Thank you.